Hi, my name is Greg Drillin, and I am a research fellow at the University of Sussex. And today I want to take you through some of the work funded by the Motor Neuron Disease Association that I am doing to help find new potential diagnostic biomarkers for amyotrophic lateral sclerosis. At the beginning, amyotrophic lateral sclerosis, or ALS, can present with a wide range of symptoms, which may include muscle weakness and movement problems, difficulties with speech and swallowing, or breathing problems. However, when patients come in for a diagnosis for ALS, this can be difficult. Only 20% of all cases have known genetic markers of the disease, and with no simple diagnostic blood tests at the moment, and a wide range of symptoms possible, diagnosis for the disease is currently a process of elimination of other potential causes of the symptoms. This results in diagnostic delay. Currently, this diagnostic delay can take up to a year. That is a full year between the onset of symptoms and confirmation of the disease. This means not only is there prolonged uncertainty for the patients and their families, but there is also a delay in prescribing suitable treatment and enrolling into promising clinical trials. This all results in a poor standard of living for patients and their families. However, with an improved diagnostic process, this could shorten the time for diagnosis, improving certainty, allowing earlier treatment, and may improve clinical trial success. So what could this improved future diagnostic process involve? We believe that this could be through a simple blood test, specifically looking for a class of molecule called RNA. And if we can identify which specific RNA transcripts are dysregulated in ALS patients compared to healthy people, we can simply screen the blood for these changes and tell more easily whether someone might have ALS or not, thus reducing diagnostic delay. And this has been the aim of our work. So our collaborators in Oxford collected blood from people with and without ALS. This blood was then spun in a centrifuge to give us serum, which removes the red and white blood cells which would obscure the detection of any biomarker. We then extracted the RNA from this serum using a resin column and into some water. With this extracted RNA, we added reagents to create what is called an RNA library. This allows us to be able to identify what RNA transcripts are present in our serum samples at the same time through the use of next-generation sequencing. This RNA sequencing machine reads the sequence of the RNA transcripts in that sample, telling us what it is and telling us how much of it is present. Once we had the data, we calculated the changes in the amount of RNA transcripts in ALS patients, whether they are increased or decreased compared to those who do not have ALS. From this, we identify the ones that are good candidates for potential biomarkers, including those that demonstrate large changes. Now, to double-check that these changes were true, we validated our potential biomarkers using a more specific technique called RT-qPCR, the same technique that is being used to test for coronavirus at the moment. So taking our RNA, we undertook RT-qPCR. For using a fluorescence dye that binds to our RNA transcripts we think might be biomarkers for ALS, we can see how much is present in our samples. Doing this, we found 7 different RNA transcripts dysregulated in ALS patients compared to healthy controls across 3 different forms of RNA, called microRNA, piRNA, and tRNA, with the names of the specific transcripts themselves on the right here. With these RNA transcripts identified as being dysregulated in ALS patients, the question became whether we can use these dysregulated RNA transcripts to help diagnose ALS. This is not straightforward though. For example, if we look at the dysregulation of one of our biomarkers, miR-21.5p, there are significant differences between the healthy controls and these ALS patients with slow or fast progressing disease, as noted by the red asterisks here. When we look at just the averages, shown by the black lines, there is decreased expression in ALS patients compared to healthy controls. However, you can already see there is overlap between the groups in terms of the amount, and with each dot representing an individual person, you can see that some ALS patients have more miR-21.5p than healthy controls, and some healthy controls have less miR-21.5p than ALS patients. So while differences between groups may be present, individual changes could be obscured simply because there are differences between people, which is then compounded by the heterogeneous nature of ALS. So the question becomes, how can we use these dysregulated RNA transcripts to help diagnose ALS? Evidently, using one of these RNA transcripts as a biomarker is not a good idea, so we should use multiple RNA transcripts as biomarkers. But how do we do this? Do we require all of them to show changes, or just a majority to diagnose ALS? Both of these are quite crude ways of determining this. An alternative method, though, is to use statistical modelling, which is more refined, and the one that we have used is called a random forest model. Using a random forest model, we make a decision tree, and at each branch point, we ask how much of this RNA transcript biomarker is there in the sample, and follow the branch that matches our sample. We continue to do this until we have used the data, and at this point, it can tell us whether the sample has come from an ALS patient or not. Now, to improve this model, we do it many times, making many different trees with different decisions at different points with the data to remove any potential bias. And sometimes these different trees will classify a sample ALS or sometimes not. After this, we simply see what the majority of the trees say, and that becomes the classification of that sample, which in this example is an ALS sample. So when we do this with our samples from Oxford, we are able to get a classification accuracy of 93.7%. That is 74 of the 79 samples we looked at were correctly classified as being from an ALS patient or not. However, as we train the model with the same data we tested it with, which inherently gives us a high accuracy, we obtained 157 additional samples from London and Germany to test this model. Looking at the same 7 RNA biomarkers in these new samples, we got a lower classification accuracy of 73.9%. We expected differences to exist between people from different places though, so this is encouraging, but we are now working to get additional biomarkers to help improve this accuracy. Nonetheless, in summary, we have identified a number of RNA transcripts that show promise in being a diagnostic biomarker when combined together to classify ALS samples. With some further work, we hope that this could be used to help diagnose ALS patients and help reduce diagnostic delay. So on behalf of Majid and the team here at the University of Sussex, we would like to thank our collaborators at the Brighton and Sussex Medical School, the University of Oxford, Queen Mary University of London and the University of Ulm. And of course we'd like to thank the Motor Neuron Disease Association, who without none of this work would have been possible.